Hello, my name is Annalise Schweickart. I am on the bioinformatics team at Harmonic Discovery, and today I'm going to be presenting my project entitled Delineating Drug Class and Target-Specific Adverse Events of Kinase Inhibitors. As background on kinases and kinase inhibitors, there are over 500 protein kinases in the human genome. These are proteins responsible for the phosphorylation of different biological molecules, and their dysregulation has been associated with many diseases, including autoimmunity and cancer. Therefore, kinase inhibitor drugs have been a successful drug class for fighting these diseases. However, kinase inhibitors are promiscuous, hitting on and off-target kinases to various degrees due to their structurally similar active sites. In the figure on the right, each panel represents the binding profile of a kinase inhibitor, with its on and off targets labeled by a red circle. Kinase inhibitors exhibit clinically relevant side effects that are at least partially due to the inhibition of their binding profiles. As more kinase inhibitors have been approved, we have seen reports of these adverse events rising exponentially, emphasizing the need for better and safer kinase drugs. So at Harmonic Discovery, we are trying to build those safer drugs by designing small molecules that can avoid adverse events causing kinases. However, to do this, we need to know which events we have the ability to prevent, as the associations between specific kinases and the adverse events they cause is limited. Therefore, the research aim of this project is to delineate adverse events that arise in kinase inhibitor administration, investigating if they are potentially due to the drug class itself, the chemical structure of the small molecule kinase inhibitor, or if they are kinase specific and thus applicable to our small molecule design. We first explored the adverse event space of kinase inhibitors using the FAERS dataset. This is the FDA Adverse Event Reporting System, and it contains physician-reported adverse events that coincide with drug administration of their patients. In this plot, each point is an adverse event in FAERS. The x-axis defines the proportion of kinase inhibitors coinciding with that adverse event, and the y-axis defines the proportion of non-kinase inhibitors coinciding with that same adverse event. From this plot, we see that for the most part, there aren't many kinase inhibitor-specific adverse events, as the proportions between kinase inhibitors and non-kinase inhibitors don't diverge to a great extent, save maybe some of these below the line of equal proportions that seem like they could be kinase-specific. In our aim to delineate kinase inhibitor adverse events, we can zoom in on the top right corner of this plot to look at the most frequent adverse events in kinase inhibitors. These events are occurring in over 40% of kinase inhibitors, and as the kinase inhibitors in the FAERS dataset have vastly different binding profiles, we conclude that these are probably not attributable to a single kinase and seem to be drug-agnostic adverse events. Next, we wanted to look at the potential role of chemical structure in kinase inhibitor toxicities. Now, there are many toxicities that are caused by molecular structure, so we decided to focus on one, which was neurotoxicity, as there is a clear role of molecular properties that allow a drug to cross the blood-brain barrier and cause neurological events. So, the traits of these small molecules that cross the blood-brain barrier are as follows. First of all, they have fewer hydrogen bond donors. Second, they have low topological polar surface area. And finally, they have a low molecular weight. We compared the molecular properties of these non-kinase inhibitor drugs that cross the blood-brain barrier with kinase inhibitors that show neurotoxicity in trials, as well as kinase inhibitors that target kinases with known neurotoxic properties. Finally, we compared this to a random kinase inhibitor set to see if the kinase inhibitors that have neurotoxic events differ from the rest of kinase inhibitors. Along these metrics, this is hydrogen bond donors, TPSA, and molecular weight, we found that neurotoxic kinase inhibitors have more chemical similarity to other kinase inhibitors than the drugs that cross the blood-brain barrier. This would indicate that potentially the neurotoxicity caused by these kinase inhibitors is due to target, on-target, and off-target effects rather than the molecular structure of the small molecule. Finally, to investigate the relationship of inhibiting a specific kinase with an adverse event, we combine two datasets. First is the FAERS dataset, which I mentioned earlier, and we filtered it down to kinase inhibitor reports. For each kinase inhibitor adverse event pair, we gave a reporting odds ratio score, which represents the odds of a certain event occurring with a kinase inhibitor, compared to the odds of the same event occurring with all other drugs in the FAERS database. Second, we used a neural network algorithm to predict the kinome-wide binding profiles for each kinase inhibitor in the FAERS dataset. This gives us a binding value for each kinase inhibitor to all kinases that we have on file. We then combine these two matrices, as seen on the right here, to create a kinase adverse event association score via the kinase inhibitors. To assess the significance of these associations, we repeated this matrix multiplication thousands of times with random binding profiles to create a null association distribution against which we could calculate an empirical p-value for the observed associations. Our method resulted in just over 1,000 kinase adverse event associations, but for the sake of time, I'm going to focus on just a few. The results I'm showing here highlight some well-known kinase toxicity mechanisms established by literature, including cutaneous toxicities caused by the RAF family's inhibition, hyperglycemia caused by the PI3 kinase family's inhibition, and acneiform rash caused by the inhibition of the EGFR family. We also found associations that are supported by literature in kinases that have no currently approved on-target kinase inhibitors, specifically the vasomotor conditions caused by the EPH family. This association was recovered purely via off-target effects. More broadly, as is evident by the figure here, the significant associations are common to families of kinases rather than single kinases. This is consistent with other research at the company that has shown that kinase families cluster by binding pocket similarity, meaning small molecules designed for one kinase in the family are more likely to hit other members of that same kinase family. So here we're showing a t-SNE of the binding pockets, and it is clustering by the kinase family from which it comes. This informs drug design in that when we want to avoid certain adverse events, we must keep in mind that these adverse events are probably occurring across the family because our small molecule will be binding to more than just one kinase in that family. In conclusion, when designing small molecules to avoid adverse events, we need to take into account that there are many adverse events attributed to kinase inhibitors that may not be due to a specific kinase binding and are drug agnostic, that kinase target inhibition may be more to blame for neurotoxicity than kinase inhibitor chemical structure, and so we don't need to focus on the chemical structure when trying to avoid those adverse events. And finally, that kinase-specific adverse events are largely family-based, and so if we want to design a molecule that will avoid that adverse event, we need to make sure that it avoids binding to the entire kinase family. Thank you.